Hi, so as Shiva mentioned, I'll be presenting on the dual responsive biohybrid Nutribots for active target delivery. And this was done by these amazing researchers listed here. Please don't make me say their names. So we'll first look at what's we first look at the introduction to the Nutribots and the construction of them, the controllable motion of the magnetical activated Nutribots, and the collective motion of the swarm of the Nutribots. Then we will look at the chemotactic dynamics of the Nutribots when they are exposed to chemokine gradients. Then we will look at how they actively penetrate the blood-brain barrier, and then how they deliver the drug. And lastly, we will look at the dual responsive dynamics of them and then the challenges. So first, the brain is one of the most precious parts of the most living things. And because of this, it is naturally protected against physical injuries with the help of the skull, the cerebrospinal fluid, and the protective membranes, which are called meninges. So the other protective elements there is the blood-brain barrier. And as the name suggests, this is the barrier between the brain's blood vessels and the cells and the other components that make up the brain tissue. But in the skull, the cerebrospinal fluid, they protect against physical damage, but the blood-brain barrier provides a defense against disease-causing pathogens and toxins that may be present in your blood. So this is generally very effective at preventing unwanted substances from accessing the brain. And this has a downside because when you want to transfer drugs to treat brain cancer, it is going to stop that. But one way that you can possibly transfer the drugs is to trick the blood-brain barrier into allowing the passage of the drug, which this is called the Trojan horse approach. And this is where the drug is fused to a molecule that can pass the blood-brain barrier via a transporter protein. And this is basically the same method that will be used by the Nutribots. And the Nutribots, they are tiny robots that can carry the cancer drugs through the blood-brain barrier without setting off an immune reaction. And for many years, the medical scientists have sought different ways to deliver drugs to the brain to treat conditions such as brain cancers. But since the brain is protected by the skull, it is extremely difficult to inject them directly. And you also cannot inject them through the bloodstream because it is blocked due to the blood-brain barrier. So that is not an option. So we will look at what the scientists came up to solve this. So they use a defense cell type that's, oh, sorry, the volume's loud. So they use a defense cell type over here that naturally passes through the blood-brain barrier to carry drugs to the brain. And to build these robots, they first used the iron oxide to form a magnetic nanogel, and this functioned as the base of the delivery system. So this is the nanogel here that it formed. And these nanogels, they contain paclitaxel, which is a chemotherapy medication that is known to be active against gliomas and various brain metastases. So they then coated this nanogel with a surrounding membrane that was derived from the bacteria E. coli. And this gave it the appearance of a dangerous pathogen. So this outside layer, it also prevents the leakage of the drug, which is very important. And then finally, they expose the coated nanogel to the neutrophil. And the neutrophil is a type of white blood cell that fights against infection, and to ensure that it would be absorbed and disguised during its journey from the bloodstream into the blood where they would not attack. So over here, you can see it's being coated. And then, yeah, the neutrophils. And this is the Nutribot here. So when they are being transferred, I will explain all of this later as well. So they inject it into the tail and they use magnets, this rotating magnetic field here, to carry the Nutribots all the way up to the brain. So it goes along to the bloodstream and goes all the way to the brain and then gives out the drugs to fight brain cancer. So now we will look at so this is the vision-based magnetic navigation system, and this consists of an optical microscope for real-time visualization and a 3-orthogonal Helmholtz-Colley pair system that is capable of generating uniform magnetic intensity. So this is just basically the rotating magnets that they use to carry the drugs along the bloodstream to the brain. So when they combine the real-time position tracking, the autonomous path planning that they did, and the vision-based feedback, the system enables self-navigation of the Nutribots along a program path that they had. And excuse me. So under the control of this rotating magnetic field, the Nutribots were actually able to reach a speed of 16.4 micrometers per second. And this was about 50 times faster than the speed of the natural neutrophils. And this is this magnetically activated navigation against the flow of blood is very essential for the Nutribots in order to accomplish the various tasks in the body. And to investigate the motion against the direction of the flow, they build a model of the blood flow using microfluidic filled with fresh blood that was applied. And this is where they saw that it was 50 times faster. So you can see in this video here, it shows the actual motion with the parts that was planned, the program parts, and using the yeah, this is all the flow of it. And this is the control one where they use the actual magnets to move it along the bloodstream. So now, okay, sorry. Yeah, so now we will look at the collective motion of the swarm of the Nutribots. So in order to satisfy the biomedical uses, it's very important to have the collective motion control of the Nutribots, because this would provide the functionality and the pathway to achieve very important tasks and real-time visualizations for imaging technologies in vivo. So they would be able to monitor it and make sure it's doing exactly what it needs to be done. And because the magnetic rotation of the Nutribots on the substance causes a local asymmetric flow, the adjacent rolling of the Nutribots are expected to assemble into a swarm due to the interaction of their local flow. So that's in figure 4A over here. You can see how they assemble with any direction of the motion. And there's a time lapse, which I will show just now. It shows the interaction behavior of the 4 Nutribots on the substrate upon an elliptically polarized RMF with a frequency of 15 Hertz. So I will show that now. So basically, you can see one. So this is one of the rolling Nutribots over here. And you can see the rest of them coming to each other to form a swarm, which they should. And yeah, they join in and they form a dynamic chain over here, which is the swarm of the Nutribots. You see more joining. So now we're going to look at the chemotactic dynamics of the Nutribots when they are exposed to the chemokine gradients. So when they're exposed to it, there's a piece of agarose hydrogel over here that was used as the chemokine source in this video over here, and then this time lapse is going to show how the Nutribots move towards the hydrogel region, which that it indicated that the chemotactic behavior of the Nutribots. So they move towards the brain, early brain cancer cells that they had used in order to actually release the drug. So they all accumulate. Well, most of them had accumulated there, which was really fantastic results. So these three over here, if you can see the kind of slightly gray ball, are the areas where they accumulate into. And now we will look at the dual responsive dynamics of the Nutribots. So it's very important to have this dual responsive Nutribots because they would be able to accomplish effective movement in real blood cells, as it is shown over here in this blood cell in figure A. So to do this, they actually got a section of the blood vessel that was freshly dissected from the aorta of a rat, as shown in figure 6A over here. And the motion of the Nutribots was observed in this blood vessel. The dual responsive motion, so the dual responsive motion process of these Nutribots that are shown in 6B over here. So the dual responsive motion that's shown in 6B over there, and the movie that I'll show just now, they were basically added into the blood vessel, and a drop of chemokine solution was injected into the dissected blood vessel to the model of the inflamed sites. So first, the Nutribots, they roll to the chemokine source, and this was indicated by the red trajectories, which I'll show in the movie just now, or also you can see the red trajectories here in B. And after that, when the inflammatory substance was turned off, the Nutribots, they began a continuous chemotactic movement, which is shown by the blue over here. And this dual responsive movement, it basically shows that after the magnetic propulsion, so after using the magnets to basically move them along the bloodstream, it still had autonomous chemotactic capability. So it's able to do what it needed to do, and it had the freedom to do it. So this is the video showing how the movement of it in the blood vessel. So the last thing will be for the challenges. I will look at the active drug delivery. So this is the actual delivery to brain cancer cells in the mice that they have used. So they tested whether the bots could treat the brain cancer. So first they injected the glioma cells into the mouse, into their brains, as you can see in A over here. And then after 10 days, they performed surgery on some of the mice in order to remove a portion of the tumor in order to boost the neutrophil actin inflammatory signals. They then injected the Nutribots into the tails, if you can see in this one over here. And in another subset of mice, they used the rotating magnetic field in order to direct the Nutribots towards the brain. And using an MRI, the team found that more Nutribots accumulated around the gliomas in mice that were treated with both surgery and the magnetic field when compared to the mice that were not exposed to the magnetic field at all. and also to the mice that didn't undergo any surgery, and to the mice that received neither the magnets or the surgery. So the double treated mice, the ones that had both the surgery and the magnets, this also survived longer. So it is surely success that it was able to actually transmit the drugs to the brain cancer cells and actually help them survive longer. And it did penetrate the blood-brain barrier and enter the glyma tissue. So all of the Nutribot treated mice that survived longer compared to the animals that were treated with an injection of just saline or paclitaxel. So this indicated that the Nutribots could still deliver drugs across the blood-brain barrier when they have a response to a very weak inflammatory signal or a strong inflammatory signal without magnetic propulsion. So it's not needed, but the magnetic propulsion definitely helps boost it. And now we will look at the challenges. So this is just a first draft of the research. It's still very new, so a lot still needs to be done to have it being used out there. So one of the challenges is that they do need to improve the amount of microbots that make it to the tumor, because I believe only 40% of it made it to the tumor. And they need to make sure that the Nutribots deliver the drugs successfully and completely. And the other thing is that they also need to think about how to transport multiple microbots in a controlled manner and deliver them to the target location. Because although we saw that 4 of them, or 5 of them, they were joined into the swarm, it still was not completely, like all of them, so only 40% of them went. So we need to find a way to be able to transport all of them. And that is it for my presentation. Thank you. And any questions?
Thanks, Gabby. So, yeah, I mean, of course, I don't know much about the area, but when you said Nutribots, from what I see so far, there's no active components in the bots themselves, right? It's just the magnetic. No. It's just the fact that it's has a magnetic shell, right? Is that the case?
Yeah, it's just
Okay, so any reason why they call it a bot then? If it's just,
well, it's they did use nanobots, but they cased the nanobots into the nanogels and into the neutrophil. And they did program a part, so it still has some of the robotics capabilities, but it is mostly the neutrophil that is doing the work to bypass the blood-brain barrier, and the magnets that is carrying it across.
okay, and and so the motion is just controlled by an external magnet, basically, right?
Yes.
Okay. I'm just curious, so is this something you're doing research in or just had an interest in?
Oh no, I just had an interest and I wanted to know how nanobots work. But I initially didn't think, well, yeah, I knew it would have a lot of biology, but I didn't know it was going to be about 80% biology based. Because I wanted to know the internal workings of a nanobot in the beginning. And I thought the research was really interesting and cool, so that's why I looked at it.
Okay. All right, thanks. No more questions from me.